First of all, I think the AMP there, the A is not for American, but for association. And so this organization tries to roll out on a global base all types of diagnostics, starting from microbiology over virology, molecular pathology, genetics, till prenatal diagnostics. And so the idea is to cover this worldwide. Therefore, the next global meeting will be in Korea, for example. This is somehow the goal of this group, to roll out the idea of molecular testing in all the different fields worldwide. We have to divide it, I think, into, first of all, the approved drugs. So if you think on targeted molecular pathology, so there basically we have only the RAS test, but in the new guidelines you also find BRAF and MSI, so microsatellite instability. So they are the markers which will somehow be involved in molecular diagnostic for the next time. And of course then there is a more research or translational field where we are thinking about resistance mechanisms, especially for anti-GF receptor targeted therapies. And of course, but this also relates to other tumors, is how to translate our knowledge into molecular tumor boards. So this means patient after last line who now need a therapy, who are usually a little bit younger in life, so let's say younger than 55 years. More and more genes show up to be important. But on the other hand, there is not such a big group, so I think it will stay on a panel level, so we don't need whole exome or even whole genome sequencing in the next future. What is important thing is that we make it digitalized, so we need molecular barcodes or unique molecular identifiers in the system to count genomes, which is especially important if it goes to liquid biopsies where we do not have a morphological correlate and we cannot determine the amount of tumor cells which are in. So we need a counter, a molecular counter for cancer, for tumor DNA. We can introduce a new bioinformatics, so we can draw out of the genetic data we generate nowadays already additional information, so like the specific background for a single patient, or that we have a chemical deamination of cytosins resulting in wrong Ts, for example, all these things. And of course, a very important thing, especially if it goes to next generation sequencing, is how to assort the results with clinical impact. So what to do with the result and what can we do for the patient. And there we need a lot of bioinformatics, which are currently, I think, generated or invented for that kind of research. Many companies develop, especially in the field of bioinformatics, but also in the field of diagnostic, in parallel many things. And on the other hand, it is somehow very difficult to integrate the knowledge which comes from universities or bigger centers like MD Anderson or Memorial Sloan Kettering Institute, this information into the commercial products, because simply they are missing usually there. But usually colleagues who work at these centers have a lot of expertise, and this is a little bit a pity that these kind of informations are missing. And I guess this would be a great thing if you could make a common product out of it and do not get rid of, let's say, all this impetus by going in so many different directions, which have all the same goal in the end. Yes.